Okay, so for this video, I just wanted to go over some misconceptions with chromosomes and chromosome numbers, what they look like, the definitions. So just so you guys don't get tripped up in coming topics. So first off, you might have noticed that chromosomes look either like this or like this. So both of these are actually one chromosome. There's one chromosome and there's one chromosome. What makes one chromosome is one centromere. So the centromere is just essentially what's in the middle of the chromosome and what's keeping it together. So see how in here there's one centromere, and here there's also one centromere. That essentially just means that both of them are one chromosome. So if I were to tell you how many chromosomes are in here and in here, then you would say that these are two chromosomes because there are two centromeres. And here you'd also say there's two chromosomes because there's two centromeres. Okay, so that's the hardest part that students get tripped up on, is the chromosome number. Just know that the amount of chromosomes is dictated by the amount of centromeres, basically. Okay, now we're going to introduce a new term called chromatid. Actually, it's not new, but I'm just going to introduce it here. Chromatid. So remember that what makes a chromosome is one centromere. Chromatid is just one copy of DNA. So one chromatid is exactly like this because it's just one copy of DNA. Now, when it goes to this form, it's still one chromosome because there's still one centromere in each, but now there's two copies of it. So see how there's one copy right here and there's also another copy right here. So those are two copies of the same chromosome. So these are two chromatids. So always remember, this is what one chromatid looks like. This one copy. So if I were to tell you how many chromosomes there are here in this set, you'd say there's two chromosomes, right? Because there's two centromeres. But you would also say there's two chromatids because there's two copies. Now if I were to ask you, let's say like this, and I'm not color coding anything here, by the way, I just randomly chose blue from here, just put it to black so you're not confused. Okay, basically it's still two chromosomes because there's two centromeres. But if it asks you how many chromatids there are, then you would say there's four because see, one, what the heck is that? There's one here, there's two here, there's three, and there's four. So there's four copies of DNA in total, which means there should be four chromatids but two chromosomes. Now this is essential in understanding exactly what happens during mitosis and meiosis. This is super important. Now, okay, so I just want to state quickly that humans are diploid organisms. Diploid just means there's two sets of chromosomes, one from our mom, one from our dad. So remember that we make gametes, right? And essentially we get one gamete from your mom, one gamete from your dad. Obviously from your dad is a sperm, and from your mom it's an egg. When it comes together you make a zygote, And essentially we are diploid because of this. But when we grow up, get to sexual maturity or get to puberty and make our own gametes, then we can make our own gametes, right? And when you make our own gametes, this process is called meiosis. But when we're just growing, so let's say growing, repairing, so those sorts of things, then we maintain that copy. So this is mitosis, and we still maintain that 2n sets of chromosomes. So this just means we get one set of chromosomes from our mom and one set of chromosomes from our dad. And remember that in each one there's 23, and equals 23 from your dad and equals 23 from your mom. So in total we should have 46 chromosomes. So in humans we are 2n, or so we're diploid organisms, and we have 46 chromosomes in total and 23 pairs of them. Okay. So now let's go over mitosis. See why the 2n that we start with when we're zygote is maintained when we go through mitosis. So the product is 2 cells. That's still 2n. And we're gonna look at how exactly that's the case. I feel like the organic chemistry tutor right now. Okay, so mitosis, as we have learned in the notes, the acronym for it is PMAT. Now, before mitosis and meiosis, there's what's called an interphase. So cell goes through interphase 90% of the time. Cell is in this stage of cell cycle 90% of the time. So you do have to know the details of it. So for example, remember there's the G1, which is growth, S phase, and G2 before it goes to mitosis. But the main important thing that you have to know here is that an interphase, the chromosomes replicate. There we go. Chromosomes replicate. Or in other terms, DNA replicates. So it's 1 copy to 2 copies now. But remember from earlier where this is essentially 1 copy of your chromosomes. But when it duplicates or replicates, this is what happens. So this is replication. This is happening in interphase. So when it's replicating in interphase, the chromosomes, which look like this, which is 1 copy of our chromosome, ends up looking like this. So the main difference between the two is that they're exactly the same chromosome, but this one is just 1 copy of chromosome. This one is 2 copy of the same chromosomes. So when it's the same copy, and there's 2 copies and they're side by side and they're the same from the same DNA, I mean they contain the same DNA, but there's just 2 copies of them, which is exactly like this. So this is what's called a duplicated chromosome. These are what's called sister chromatids. So sister chromatids are same copy of DNA, but there's just 2 of them. Okay, so during interphase, the big kind of event that happens is that your chromosome duplicates from this to this. Now remember that this one is still 1 chromosome because there's 1 centromere, and when it duplicates, see how it's still 1 centromere? That means it's still 1 chromosome. But See how this one is one chromatid because it's one copy? Now, when it duplicates in interphase, there's two chromatids, which means that your chromatids duplicate, but your chromosome number stays the same. Okay, so that's kind of the basic foundation of what you should know when it comes to mitosis and meiosis. If you don't understand this, then essentially you're just gonna get messed up in each kind of question. So yeah, now we're gonna go through the actual steps of mitosis. So remember in interphase, I'm just gonna write it out again. So in interphase, we go from here to here. Now I just wrote out one, but for humans, there should be 46 of this, right? So in humans, we have 46 chromosomes. So we start out with 46 chromosomes and 46 chromatids. Chromatids, there we go. And when it replicates in interphase, what happens is the chromosome number stays the same because there's one centromere in each. It's still 46 chromosomes. But now there's going to be 96 chromatids because there's double the copy. So that's the main difference in interphase. Now what the general term to write it out as is 2n equals 46. So this refers to your ploidy, which means how many sets of chromosomes you have. And essentially we have 2 because we have 23 from our dad and 23 from our mom. Now from now on, I'm just going to start color coding it. Blue chromosomes from dad. Let's see. There you go. What the heck? And red from your mom. And remember, there should be 23 from your mom and 23 from your dad. In total, 46 when you, yeah. Okay, so that's interphase. Now remember, the stages in mitosis is PMAT. PMAT, we're gonna go through each one. So, prophase. Prophase. Draw the cell here. The main events that happen in prophase is that chromosomes condense. Chromosomes condense, which means that they essentially just pack tightly together. Pack tightly together. Another thing that's happening is the nuclear envelope starts to disintegrate. Another big thing that happens here is that centrioles, always think of the O's in the end for poles, move to poles. And lastly, spindle fibers are formed from centrioles. So centrioles essentially just kind of act as anchors for these spindle fibers. Okay, and then now we're gonna try to draw it out. So just for convenience sake, I'm not gonna draw out all 46 chromosomes. I'm gonna do 2n. Let's see. I'm gonna do 2n equals 6. So if it's 2n equals 6, it means there's 2 sets, which means half of your set comes from your mom. Forgot to color code it. Half of this comes from your mom, half comes from your dad. Which means there should be 3 red chromosomes, 3 blue chromosomes in total adds up to 6. Okay, so first let's draw the chromosomes condensing or packing tightly together. And how to draw that is that you just draw out what you normally see with chromosomes. So you just draw out the normal chromosomes. So this is what a condensed chromosome looks like. So remember, there's 3 for your father's chromosomes, and there should be 3 from your mom's. Now, if you were talking about a human cell, of course, there's 23 blue and 23 red, right? But in here, we're just doing 3 of each, so it's yeah, for convenience sake. Okay, so again, let's look at the numbers. We start out with prophase or mitosis as 2n equals 6. If you count out how many chromosomes there are, there's 1, 2, 3, 4, 5, 6. Because remember, it depends on the centromeres. So there's 6 centromeres, there should be 6 chromosomes. It's 2n because there's 2 sets. The one set comes from your mom, one set comes from your dad. Okay, and then let's see, nuclear envelope starts to disintegrate. So we're just going to draw it out like this, where the envelope around it just doesn't look like it's fully formed. Centrioles start to move to poles. So centrioles just kind of look like this. They're 2 proteins like that. It's kind of like a cross shape. I'm going to draw it out as, let's see. I'm going to do orange. Okay, so it's not fully at the poles yet, but it's starting to go to the poles. So just draw it out kind of close towards the poles. There we go. And the spindle fibers also start to form. So these are here. So they're not attached yet, but it's starting to, some of it is attached, some is not, but it's just starting to form from the centriole. Okay, so that's your prophase. Next is metaphase. Now it's easy to remember metaphase for the letter M. Think of the letter M for middle. And this just means that chromosomes, actually I'm going to do a distinction here. All chromosomes are lined up at the middle. And another way to remember prophase is prof means begin. So that means that when this phase happens, that's when mitosis happens. Okay, so all chromosomes are lined up at the middle. What else? All spindle fibers are connected to the chromosomes. Okay, so let's draw that out. Okay, so at this point, nuclear envelope is gone too. So it started to disintegrate in prophase, but nuclear envelope is just like gone. Okay, so let's draw this out. So there should be 6. It says here all chromosomes are lined up at the middle, so that's what we're going to do. We're going to draw out all 6 of the chromosomes lined up together. 1. I'm just going to leave out space for the red. There we go. 3. That's actually... Oh, I forgot there's no envelope anymore. 1, 2. Okay, here, and then I'm going to draw out the red chromosomes, which represent your mom's chromosomes. Okay, now I know that doesn't really look like it's all in the middle, but it should be in the middle. Just gonna write this out just to show that it's in the middle. There we go. And again, it doesn't really show, but there should be 6 centromeres there, which show that there's 6 chromosomes. Okay, and then we're gonna draw out our centrioles. And then we're going to draw out the spindle fibers, and we're gonna show that it's connected to all of the chromosomes. There we go. So that's what I should see during metaphase. Next is anaphase, and again, this one's pretty easy to remember. It's A for away. Anaphase. Here we go. A4 away. Okay, this one is a super important distinction. Sister chromatids. And just to clarify again, remember, sister chromatids are just chromatids that contain the same DNA. They're just two copies of each other. So these would be sister chromatids, this one and this one. Sister chromatids are separated to each side of cell towards their poles, to their respective poles. Okay, so I'm gonna draw it out. Right, so this one's the hardest to draw, but basically I'm just gonna show that the chromatids are now how do I draw this? Okay, here we go. Like this, yeah, there we go. I'm just showing that the chromatids are split apart from their sister chromatids. And then we're gonna do the same for the red. That doesn't really show look like it. Oh. There we go. There we go. And then on this side, we're going to do the same thing. So see how it's now split? I'm just showing that it's a V because it's being pulled away. I'm just showing inertia. Okay, and then at the poles, there's still your centrioles. Centrioles. And then it's connected there. And those chromatids become their own chromosomes. Because remember that what makes a chromosome is one chromosome equals one centromere. So when it's split apart here in telophase, so the next step of mitosis is telophase. Telophase right here. What essentially happens is nuclear envelope starts to reform in each of the cell and cell starts to elongate. Okay, so what we should see is something like this. So see how the cell is starting to elongate into two different ones. So we're gonna and also the spindle fibers are gone here as well because they did their duty. They separated the sister chromatids, which means they're pretty good. And remember how I said that in mitosis this is maintained. All the chromosomes that you need is still maintained because remember the sister chromatids are the same exact copy of the same DNA, which means you're not losing any DNA information because this side is the same DNA information as this side is the same information as this side, which means that each cell essentially gets the same amount of information. It's just now unduplicated chromosome, which is a chromatid. Okay. So now I'm gonna draw out that the poles are starting to kind of move away from the center. They're not in the poles anymore. There we go. Just ignore the yellow highlight. Spindle fibers are gone. The nuclear envelope is starting to reform again, so I'm just going to draw something that looks close to done. And each cell should have 6 chromosomes where we started. So this side should be here. So we're just going to draw it out. Should be 1, 2, 3 chromosomes from our mom. There should be 1, 2, 3 chromosomes from your dad. Now that sounds familiar, right? Because that's what we started out with. The only difference is that this one is duplicated chromosomes, this one is unduplicated. But it's still 6 chromosomes in total, because remember, what makes a chromosome is the centromere. So if you count the number of centromeres, there's 1, 2, 3, 4, 5, 6 centromeres. And we started out with 6 centromeres here at the top as well. The only difference is that the chromatid number has been halved. So we started out with, if you count all the chromatids here, there should be 12 chromatids at the start. But at the end of mitosis, there should be 6 chromatids. But remember, chromosome number stays the same. Okay, so on this side, there should still be 3 from your dad as well. And then 3 here. There we go. There we go. Right. So we're done meiosis. We're done mitosis basically. Because see how the nuclear envelope has formed here? Which means that they're technically within their own cells now. But the process of it splitting into 2 separate cells has to do with the cytoplasm here. And that's the process of cytokinesis. Process of splitting cytoplasm into 2 pieces. Okay, so we split the cytoplasm into 2 pieces, which means that at the end here, you should get 2 separate cells. You already divided it. Your nuclear envelope looks good. And each side should be 2n equals 6. 2n equals 6. And there we go. And if you look at your notes, you would know that the cell cycle happens around 50 times until it dies. So after the end of this process, cytokinesis is going to go back to interphase. And during interphase, the whole replication process happens again, right? So what happens to each one of these is that the chromosomes start to double like this again. And then essentially it's going to go through the same process. So it's going to go back to mitosis. Okay, so this is kind of the amount of work. Oh, actually, I forgot to mention that the centrioles still exist. Always draw your centriole. So centriole is always going to be in the cell. It's just that during prophase, it starts to go to the poles. Your metaphase is at the poles, and then just draw out your centrioles here. The only ones that disappear are your spindle fibers and your nuclear envelopes, essentially. So spindle fiber starts to disappear in telophase. Okay, so this is what I would expect if I were to give you 100 on like a quiz or something. This much kind of drawing, this much detail. Yeah. Okay, so now we're gonna look at what the difference is between meiosis and mitosis. But this shouldn't be too hard for you now at this point, because we know that mitosis, we've already established that mitosis starts out from 2n to 2n. And here I just used a 2n equals 6 example. So see how we started out as 2n equals 6 here, and it ends with 2n equals 6. Because remember, there's still 6 centromeres in each. The only difference is just that the chromatid number has been halved. Alright, and again, I really don't want people to get confused by this. The 2n just means 2 sets. One from mom, one from dad. Which means that if, for example, you had an organism that was 2n equals 12 in total, it means that half of their chromosomes, which means they got 6 of their chromosomes from their mom, they got 6 of their chromosomes from their dad. Does that make sense? Okay, so we're at the 30-minute mark. Shouldn't be too bad. Okay, so now we're gonna move on to meiosis. And this is when you go from 2n to n. Meaning you go from 2 sets of chromosome to 1 set of chromosome. And meiosis happens 2 times. There's meiosis 1, which is essentially the exact same thing. There's prophase 1, metaphase 1, anaphase 1, and telophase 1. And then meiosis 2, same thing, prophase 2, metaphase 2, anaphase 2, and telophase 2. And between these, there's cytokinesis. Remember, there's a splitting of the cytoplasm. And then there's also cytokinesis here. And what you end up with is 4 daughter cells. So remember, in mitosis, you end up with 2 cells here, and then the chromosome number is maintained. With meiosis, you end up with 4 daughter cells. Each is N. So if I were to say, so this is something that you should kind of train your brain to do. When you see something that an organism is diploid, for example, I'll say this organism is 2n equals 14, right? When it makes its own gametes through meiosis, you should end up with n, which means half of this. So each 4 daughter cells should end up with n equals 7. So that's kind of how you cross-check your the amount of chromosomes at the end. Is you see, okay, so this organism is diploid. It starts out with 14 chromosomes. After it makes its own gametes, then is going to have n equals 7 in each cell. Okay. Same thing with mitosis. Meiosis cells goes through interphase and then it duplicates, which means it should start out as this duplicated chromosome like this. And again, we're diploid, right? Which means we have 2 sets of chromosomes, 1 set from mom, 1 set from dad. Which means that for a human, half of it is from mom. Half of your chromosomes come from your mom, and half of your chromosomes come from your dad. And again, I'm gonna start to color code these into blue and red. So let's see, how should I start this? Okay, let's just start out with, I'll just start out with explaining what a homologous chromosome is. Actually, I'll do this video in 2 parts. I'll stop this video and I'll continue with the next video, starting with explaining what a homologous chromosome is. So